Okay, so welcome to our vodcast on cell transport, the movement of cellular materials. Now, what we're going to talk about is how materials get into and out of the cell. So in order for that to happen, they have to cross a certain organelle that forms the outside of that cell. And that cell organelle is called the cell membrane. Okay, if you remember from our lessons on cell organelles, the cell membrane controls what enters and leaves or goes in and out of the cell. Now, the first type of transport we're going to talk about tonight is called passive transport. Now, passive transport is called passive transport because it does not use any energy. So if you think about it, if you're passive, you're not doing much of anything, you're not very active, and if you're not active, you're not using energy. And the reason why passive transport does not use energy is because they move along what we call the concentration gradient. You have to remember, a concentration gradient is when one area has a lot of one thing, so it's a high concentration of stuff, and another area has not as much stuff. So you have a low concentration. And that's going to be your concentration gradient. And moving along with the concentration gradient means you're going from high to low. So that's your natural movement or natural flow. So the first type of passive transport we're going to talk about tonight is diffusion. So let's take a look at an example and how this all works. So first thing to remember, diffusion is the movement of gas or liquid molecules going from an area of high concentration to low to establish an equilibrium. So when we take a look at this demo setup, I have two beakers set up with dialysis tubing. The dialysis tubing is filled with colored chemicals. Here in this bag we have iodine, which is a chemical indicator, and a stain. And also we have this bag just filled with food coloring. So if you take a look, these bags are in stark contrast with the water that they're placed in because they have a lot of color molecules inside the bags. So we have a high concentration of color molecules in the bags and a low concentration in the water. So there's our concentration gradient. High in the bags, low in the water. Now over time, as diffusion states, these molecules are going to move out of the bags and into the water. And if we take a look at 60 minutes later, that's exactly what happened. All right, we can see that the color molecules left these bags and went into the water where there was no color molecules before, moving along the concentration gradient, not using any energy. So let's take a look at how this actually happened or worked.
Now, to understand how passive transport works and why molecules move from areas of high concentration to low without energy, you have to understand how the molecules move and what they do. So in the simulation above, in region A, we have a bunch of molecules that are in motion, and typically gas or liquid molecules are constantly in motion. So in this simulation, let's pretend the purple spheres are gas molecules. So when we take a look, these molecules are moving randomly, and because they're moving randomly, they're bouncing off of one another and causing these collisions. Now, these random collisions are what cause these molecules to move out into different areas. So when we talk about diffusion, simple diffusion of gases and liquids other than water, this is going to be the driving force as to why they move from areas of high concentration to areas of low concentration. So let's take a look at see what happens when we open up this wall. When we take a look here, region A has a high concentration of molecules. So as a result, when these molecules start moving, again, we're going to see those random collisions start to occur. And when those random collisions start to occur, that's when these molecules start to spread out. If you watch what happens when these molecules bounce off of each other, some of those molecules are going to be sent through that open space above that wall or the red line. And this shows or causes the movement to go from an area of higher concentration to low. Because of the random movement of the molecules and the random collisions that are occurring, these molecules are going to move and eventually start to create a balance in numbers on both sides. And this is what we call equilibrium. When equilibrium is reached, there's no linear or straight directional movement going from a high concentration to low. It's now just a matter of these molecules randomly moving, and some will go back to region A and others will go back to region B, and you establish what's called the dynamic equilibrium, which is the random movement of molecules to establish or keep this balance of numbers on each side. So that's how simple diffusion works. Just a random movement of molecules bouncing off of each other, going from areas of higher concentration to areas where there's less of them and a lower concentration.
All right, so I hope the animation kind of clarified things for you, but just to tie it in and wrap it up. In that animation, we were seeing that the molecules, such as these chemical molecules in here, were randomly bouncing off of each other. So as they randomly bounce off each other, they're eventually going to find their way through openings. And dialysis tubing has holes in it, tiny, tiny pores, so it's semi-permeable. So that random bouncing and colliding that these molecules were doing were sending color molecules out of the bag. So that's why the beaker water started to change color over time, because those collisions sent some of those molecules into the water. And not only did they do that, as the color molecules got into the water, they continued to bounce off of each other and then spread throughout the water. And that's why you don't have like one area that's super concentrated and dark in color. You'll notice that the water is pretty evenly colored. So this is one example of passive transport through simple diffusion. The other type of passive transport that we'll talk about that's pretty important is osmosis. Now, osmosis is a type of diffusion, but it only involves water. So really, the definition of osmosis is the movement of water from a high concentration to low to establish an equilibrium, or simply put, it's just the simple diffusion of water. So let's take a look at how osmosis occurs and happens in a cell.
So here we have our cell set up, and we have our cell membrane in the middle where the water molecule is going to pass through. We have our water molecules, which are the red and blue molecules that look like Mickey Mouse. And here we have our concentration gradient. Since we have more water molecules on the outside, we have a high concentration set up on the outside of the cell, and then fewer, creating a lower concentration gradient on the inside. Osmosis will move from high concentration to low, and that's what we'll see here. So as the water molecules bump into each other, they're going to find their way into openings called aquaporin, and they are going to move through those channels into the cell, trying to establish an equilibrium and a balance. Now let's take a look at how osmosis affects our cells. Now here we have red blood cells, which are the cells that carry oxygen into our body. Now in the blood, you're going to have dissolved materials called solutes, and these are going to take the place that water could occupy. So if you have a higher concentration of solutes on the outside, that means you're going to have a low concentration of water out there. If you have fewer solutes on the inside of the cell, you're going to have more water, so you'll have an area of high concentration. Because this is osmosis, water will move from an area of high concentration to an area of low concentration, which for this example, it means that the water is actually going to leave the inside of the cell, and as a result, the cell will start to shrivel up, which isn't very good because the cells need water. However, we can set up the scenario in the opposite way. On the outside of the cells, we have less or fewer dissolved substances, so we're going to have a higher concentration of water. And on the inside of the cell, we have more solutes, so we're going to have a lower concentration of water. So because of this concentration gradient, the water is going to move from the outside of the cell to the inside of the cell. And because it's going to move from the outside of the cell to the inside of the cell, this is going to fill the cell up with water, and as a result, the cell will swell up and then eventually burst.
Okay, so that was osmosis. Now our last type of diffusion we're going to talk about, or our last type of passive transport we're going to talk about, is called facilitated diffusion. Now if something is facilitated, it's being helped along. So what we have here in facilitated diffusion is the movement of large molecules such as glucose, sugar molecules, moving from an area of high concentration to low to establish equilibrium with the use of special carrier proteins. Okay, that sounds like a mouthful and it sounds like a lot, so why don't we take a look and see what happens during facilitated diffusion.
Facilitated diffusion is just like regular diffusion and osmosis. They're going to move materials from areas of high concentration to low without the use of energy. In this example, we have 5 of these molecules on the outside of the cell, and we'll call them glucose for our purposes, creating an area of high concentration, and then 1 on the inside of the cell, creating an area of low concentration. Therefore, we have our concentration gradient. In facilitated diffusion, we use large molecules, and as a result, they can't fit through the bilayer or the phospholipid bilayer of the cell membrane. So they need help. They need something to facilitate them through. And what they use is that big purple structure in the middle called a transport or carrier protein. So as our glucose molecules float around, they'll eventually randomly move into this carrier protein and fall into what's like the active site. And in this active site, what will then happen is the transport protein or carrier protein will change its shape, and then it'll send this molecule down through the cell membrane and into the cell, and is facilitating the diffusion of this glucose molecule. And as you can see, a second one is going to fall into that site again, and it'll also cause the protein to change its shape, and it'll facilitate or help along the diffusion of this molecule. And that's what facilitated diffusion is.
All right, well, that wraps up our passive transport segment of this lesson. So let's go to another type of transport. Now, the second type of transport is the exact opposite of passive transport. This type of transport is actually called active transport. If you're wondering why it's called active, well, if you think about it, if you're being active, you're using something, and that something is energy. So active transport uses energy. And the reason why it needs to use energy is because it has to move against the natural flow of things or the concentration gradient. So instead of going from high to low, like passive transport does, active transport goes from low to high. So if you think about it this way, think about the natural flow. If you're caught into a rip current, the natural flow is to pull you out to sea. But if you swim straight towards the shore against the flow of it, you're going to be burning up a ton of energy. That you're actively swimming, using up a lot of energy. So that's one way to remember what active transport is. Okay, we're moving against the concentration gradient. So let's take a look and see what that looks like. Now, our example here using active transport is a famous mechanism in your cells called the sodium-potassium pump. And this is the pump that actually helps your neurons or brain cells and nerve cells send impulses throughout your body so you can do certain things like think, talk, walk, and everything else in your day. So let's talk about how active transport works here. What we're going to do is we're going to follow this ion here called sodium to start off. The sodium is this orange shape right there, or this orange molecule. So if you take a look on the outside of the cell, that's the top here, this is the outside, we have a ton of these sodium molecules, or sodium ions. And if we look at the inside of the cell, we don't have a lot. But you'll notice that these molecules are going into the transport protein from the inside of the cell. They're not coming in from the outside. What ends up happening is this. Through active transport, we're going from an area of lower concentration inside the cell out to an area of higher concentration on the outside of the cell. And to do that, we need these carrier proteins. So these ions bond to the carrier proteins, and then what ends up happening is a molecule of ATP. So this is our chemical energy that we use for everything, will bond to that carrier protein. And when it does, it's going to transform that carrier protein and make it active, and thus release the sodium ions into an area of higher concentration. So this is why we use energy to move from areas of low concentration to high, because we have to activate the transport protein or the carrier protein. Now, when ATP gives energy, it loses one of these circles, and it's just a simple phosphate. So that's how the sodium gets out, okay? And this will help set up your nerves for an impulse to come in. And then, as a result, what will then happen is, since this channel or this carrier protein is open, you're going to have these potassium ions floating around. And as you'll notice, there's only 3 of these potassium ions on the outside, and there's a ton on the inside. So these guys want to get in. So we're going from, again, an area of low concentration to an area of high concentration. So these potassium ions bond to the carrier protein. That phosphate that's attached to the transport protein will continue to activate it, and thus open up the transport protein, sending in the potassium ions into the area of higher concentration. So as you can see, active transport is the move of materials going from a low concentration to high. Now let's take a moment and see how that looks in motion.
Now, as we've been talking about passive transport, we've been going from an area of high concentration to an area of low concentration. And this requires no energy. However, in active transport, it's going to be the opposite. So here we have what's called the sodium-potassium pump, and we have to take these Na or sodium ions and move them from an area of low concentration from the inside of the cell to the high concentration on the outside. So in order to do this, we have to go against or push against or move against the natural flow of things. So we're going to have to use energy to do this. So we use this transport protein to push us through, and as you can see, these 3 sodium ions, the Na pluses, are going to go into the carrier protein, and an ATP molecule, a molecule of energy, will bind to it, drop off of phosphorus. That phosphorus will cause the transport protein to open up, and those sodium ions will move out into the area of high concentration. So we've gone from low to high using energy. While that's open, the potassium, the letter K ions, are going to go from an area of low concentration from the outside of the cell to a higher concentration on the inside of the cell.
Okay, so that's active transport. Now the last type of transport we're going to do doesn't involve the passing materials, let's say, through the cell membrane. So it involves the cell membrane, but these materials are a little bit bigger and cannot pass through protein channels or straight through the phospholipid bilayer. So the two things that we're going to talk about, are the two modes that we're going to talk about, are endocytosis and exocytosis. How can you remember the difference between these two modes? They sound real similar and they're easy to get confused. But if you break down the word exocytosis and endocytosis, you can kind of see what the differences are. So if we take a look at the word exocytosis, you might remember the term exo means outside. Cyto means cell. Okay, so when you hear the word cytoplasm, that's just basically like saying like cell fluid or cell plasm. So exo means outside, cyto means the cell, and this means outside the cell. What exocytosis is, the release of materials stored in a vacuole that fuses to the membrane of the cell and then gets released outside. So as you can see in this diagram, we have our vacuole with materials that's stored on the inside, and this vacuole is going to move towards the cell membrane. You'll see that the edges of the vacuole start to make contact with the edge of the cell membrane and thus fusing together. And when they fuse together, the contents on the inside of that vacuole get released and go outside of the cell. So they are exiting the cell. All right, so that's why it's called exocytosis. And endocytosis is just the opposite. Okay, if you take a look at the word endocytosis, the word endo means inside, cyto means the cell. Now the definition of endocytosis is the taking in of matter by a cell by engulfing it with its cell membrane to form a vacuole. So if we take a look here at this diagram, you'll notice that these are materials that need to be taken in by the cell. So what the cell does is it causes a depression, and these materials fall into that depression. And then the cell membrane is going to enclose or engulf the materials around it. And then that gap that was open here is now closed off by parts of the cell membrane. And eventually these two walls of the cell membrane will then fuse and create this vacuole, which will then store the materials taken in by the cell. And that's endocytosis. So that's the difference between exocytosis and endocytosis. And that's going to conclude our vodcast on cell transport. Thank you for your time.